on now to session 2. Session 2 is the topic of exploring druggable pathways to treat EHE. We have 3 presenters. Our first presenter is going to be John Lamar, who's going to be presenting on the topic of uncovering new therapies and clinical biomarkers in EHE. Dr. Lamar, welcome aboard.
Well, I want to start by thanking David for the introduction and the organizers for the opportunity to participate in this wonderful conference, and also the EHE Foundation in general, as they're funding both of the projects that I will briefly talk about today. And so you already got a brief introduction about EHEs causing fusion proteins from both Kieran and others this morning. And so just to remind you that chromosomal translocations that occur in cells lead to the generation of these fusion proteins, TASCAMTA1 and YAPTFE3, which you've been hearing about. And these are the driving events in EHE. They go to the nucleus where they partner with the TEAD family of transcription factors to regulate the genes that are driving the formation and the subsequent growth of EHE. You also heard that some patients are diagnosed with an aggressive form of the disease that is very rapidly growing and ultimately deadly. But other patients are diagnosed with an indolent or very slow or not growing form of the disease. And those patients can be stable for months, if not years, with this indolent form of the disease. But if their tumors are inoperable and they can't have them removed, they're ultimately at risk for potentially progressing to this aggressive form of the disease. And so there's been a really amazing amount of progress that's been made, I think, in EHE since I've started in the field. But there are still a lot of unmet needs, and I'm not going to list them all, but I just want to mention the ones that my lab is starting to focus on. And so we really need to find more effective treatments for this aggressive form of EHE. But in addition, it would be nice to find better ways to manage and if not treat the indolent form of disease to at least prevent it from progressing to that aggressive form. And along those same lines, it would be nice if we had more effective ways to actually predict the progression of the disease from the indolent form to the aggressive form. And then once patients start treatment, it would also be good to have ways to monitor the effectiveness of these treatments so that patients don't stay on therapies that are not working for them any longer than they need to. And I'm not going to really talk about this too much today, but we're also very interested in the pain that many EHE patients experience and trying to understand the causes of that and look for ways to improve that. And I'll touch upon that at the very end. And so we're going to start by a project that's really aimed at addressing these first 2 needs. And this is work that has been spearheaded by a very talented graduate student in my lab named Sarah McMullen. And she took basically a library of 190 FDA-approved drugs. And so since EHE is so rare, the likelihood that drug companies are going to invest a lot of money in EHE-specific treatments is very unlikely. And so what we think is a good approach is to take drugs that are already approved, been shown to be safe in the clinic, and repurpose them to use in EHE patients if they are effective. And so what Sarah did was she took mouse EHE tumor cells that were generated in Brian Rubin's lab, and she treated them with 190 different FDA-approved drugs that are being used to treat other diseases, and then just looked to see which of those drugs reduced the survival or the viability of these cells by greater than 50%. She also did the same experiment in non-cancerous cells with the goal of trying to identify things that are specifically going to affect the EHE cells and have less of an effect on normal cells, which would, in theory, limit the side effects to the patient. She then rescreened the hits in a second EHE cell line, again, that we obtained from Brian Rubin's lab. And then that allowed us to identify drugs that are specifically affecting EHE and not normal fibroblasts. And so I'm not going to show you all the data, but this is a summary of the initial hits that Sarah got. And so there were 24 drugs that reduced the viability of the first EHE cell line, which are these blue bars. Most of those drugs also affected the viability of the second EHE cell line, which are the lighter blue bars. But also most of them did also reduce the viability of the normal cells. And so there was only 5 of the drugs that affected the EHE cells and didn't really affect the normal cells. And interesting to us, 4 of those 5 drugs were statins. And so statins are a relatively commonly used drug to control cholesterol levels in patients that are at risk for cardiovascular disease. They have a pretty minimal side effect profile, and patients can take them for long periods of time. So this caught our attention as a potentially very safe drug that may benefit EHE patients. And so we explored this a little bit further. Sarah took multiple different statins that are used in the clinic and tested their ability to affect the growth of the EHE cells. These are cells growing on a dish. Again, both of the cell lines from Brian Rubin's lab. And all the statins reduced the viability of cells rather dramatically in the nanomolar range, a little bit higher range in the EHE 17 line, which tends to be more resistant to everything that we do to it for whatever reason. But it still worked. Sarah went on to show that these drugs are affecting the viability of these cells by inducing a type of cell death called apoptosis, which we can measure with the levels of this protein called cleaved caspase. And so when the cells are dying, they cleave this protein, and you can measure it. And you can see that as the statins dose increases, it induces this apoptosis in these cells, and that's just quantified down here. So I don't have time to go through what was really a lot of hard work by Sarah, trying to understand what the statins are doing. I'll just tell you that statins control cholesterol synthesis in cells by blocking this enzyme called HMG-CoA reductase, which generates something called mevalonate. And mevalonate is used to generate cholesterol, but it's also important for post-translational modifications of other really important proteins in the cells. And one of these families of proteins are known to regulate YAP and TAZ. And so we predicted that maybe statins were having this effect on EHE by basically blocking the activity of TAZ-CAMPTA1 in the same way that they block the activity of YAP and TAZ. And I'll just summarize that basically that is what Sarah found. And so if she treats the cells with statins and then restores some of these different intermediates, she found that it's really the mevalonate regulation of this substance called GGPP and the subsequent activation of Rho that is what's being inhibited by the statins. And she showed that the statins reduce the levels of TAZ-CAMPTA1 in the EHE cells. And so that kind of gives us an idea of how they're working. So all of this work so far has been done in the mouse cell lines that were provided by Brian Rubin's lab, as I said. We were also fortunate now through our collaboration with Sandro Pasquale and Silvio Stacchiati, the Italian group that had generated the human-derived EHE cell line. And so this came from a patient with a very aggressive form of EHE. And so we got these cells, and Sarah repeated all the experiments I just showed you in these cells. And she again saw that the EHE cells are very sensitive to the statins. The statins are inducing apoptosis in these cells in the same way that they are in the mouse cells. And it's through the same pathway, and I'm not showing that data here, but it's the mevalonate pathway that's being inhibited, and that's important for the survival of these EHE cells. So this has all been done on cells in a ditch. And so obviously, and I'll also mention that we have recently obtained the cell lines generated in Meniere's lab, and so we're testing those now for their sensitivity to the statins. But again, all this has been done in cells on a dish, and so what we really want to know is, are statins going to benefit or prevent tumor growth in a living organism? And so for this, we turn to a preclinical mouse model of EHE, again, taking those human EHE cells that we got from Sandro Pasquale, injecting them into mice, and then once the mice form palpable tumors in about 20 days, Sarah treated half the mice with statins and left the other half untreated, and then measured the volume of the tumors over time, the growth rate of the tumors. And so what she saw was that the mice treated with the statins showed a significant reduction in the growth rate of the tumors compared to the control mice, which are the black bars here. And so at the end of the experiment, the tumors were significantly smaller, and these mice actually lived longer than the untreated mice. And so it extended the survival of the mice. And this was not dependent upon the sex of the mice, and so both the male mice and the female mice benefited from the statins relative to the controls. And so this shows that statins can extend the survival of mice bearing EHE, and that it slows the growth of this aggressive form of EHE. And so one of the things that we're interested in testing now is whether statins can also maintain an indolent EHE as an indolent EHE and prevent the progression to a more aggressive form. And that's something that we're hoping to look at in the future. The other thing that Sarah wanted to know was whether or not statins can be combined with other existing EHE therapies. And so I'll just point out right now that statins cannot be taken in combination with sirolimus. They have negative effects on each other. And so if you're on sirolimus as a patient, you do not want to put yourself on statins. And you should always check with your doctor before doing anything. But the drug that we did look at was a TEAD inhibitor. And you've heard a little bit about those in the previous talks. And so this figure right here is just showing that when you treat these human EHE cells with either a statin, you get a reduction in the viability of these cells. And you see the same thing if you treat the cells with this TEAD inhibitor. And so again, these are TAZ-CAMPTA1 driven EHE, so that the TEAD inhibitor is going to work in these cells. It's very potent. But what Sarah wanted to know is what happens when you combine these two drugs together. And so we did something called the synergy plot here, which will show you an interaction between the drugs where you get a beneficial effect of combining them above individual drugs alone. And so the red peak here tells you that combining the drugs is giving you an added benefit to the drugs alone. And so she saw this in the EHE cells, but if she does the same experiment in a non-EHE normal cell, fibroblasts, we don't see any interaction of the drugs. And so the TEAD inhibitors are still effective at reducing the viability, but the statins don't do anything to enhance that. So this enhanced effect is specific to the EHE cells. And we also looked at the genes that are regulated by the TEAD inhibitors, that are inhibited by the TEAD inhibitors, and we see that I'm not showing the data, but we see that the statins more dramatically affect the transcriptional effects of the TEAD inhibitors. And so the genes that get repressed by the TEAD inhibitors are repressed more dramatically when you add statins on top. So we think that the statins are enhancing the effects of the TEAD inhibitors, which may allow them to work at a much lower dose than they would necessarily be needed to be used in if treated alone. And this is important because these TEAD inhibitors, one of the challenges with them that's being observed in the clinic is that they have dose-limiting toxicity to the kidney. And so if we can figure out ways to give less of the drug and get the same effect, perhaps by combining it with statins, that may give a benefit and allow patients to go on those TEAD inhibitors for longer and benefit from them. So I'll kind of stop with that first part of the project and just briefly talk about a second project that we've just started that really tries to address some of these other unmet needs that that I outlined in the beginning. And so this is a project that was recently also funded by the EHE Foundation. So we're very grateful for their support of this. It wouldn't be possible to do this without their support. And so again, these tumors are driven by these fusion proteins, which drive gene expression changes, which promote the growth of the tumors. But because these fusion proteins are somewhat novel, we predict that they may have targets that are not normally being regulated in normal cells. So proteins that are being made by these cells that are not being made typically by other cells in the body. And so in some of these cells, these proteins are going to be secreted by the cells. And so this raises a possibility that we can use these TASCAMTA1 regulated proteins or YAPTFE3 regulated proteins as a way to identify biomarkers for EHE. And so some of these secreted proteins are going to end up in the bloodstream. And if we can collect the blood from the patients, we may be able to find something that's specific to the tumor and then use that to monitor things like the progression of the disease to a more aggressive form. So a more aggressive form of the tumor that's growing faster may secrete more of these proteins. So you'll see an increase in these biomarkers. And then conversely, if you have a treatment that's working very effectively to block TASCAMTA1 function or to reduce the growth rates of these tumors, you will see, in theory, a reduction in this biomarker. So it provides a non-invasive and real-time way to measure or monitor the effectiveness of a treatment as well as the progression of the disease. And ultimately, what we'd really like to do is be able to use these circulating biomarkers to predict whether or not a patient's going to respond to a disease so that they don't go on, respond to a treatment so that they don't go on a drug that's not going to benefit them and waste those precious months discovering that. And then ultimately, I think the goal is going to be to identify some of these proteins that are specific to EHE cells that are being secreted. And so this might reveal new therapeutic targets because some of these proteins are actually having an important role in the biology of the tumor. And this is where that potential direction of taking into the pain management goes, because a lot of times pain is driven by some of the things that cells secrete. And so if the EHE cells are making something that's driving or causing pain, we might be able to identify it with this approach. And so the way that we're doing this is sort of depicted here on this slide. We have a way to engineer the EHE cells so that they express an enzyme that will attach a biotin, which is a little marker, to all proteins that the cells are secreting. And that's important because then we can use streptavidin-encoded beads to basically collect all those proteins that the EHE cells are secreting and identify what they are. And so what we'll do is inject these cells into mice, allow the mice to form tumors. We can then treat the mice with biotin, and the EHE cells will start basically tagging all the proteins they secrete with this biotin. And some of those are going to get into the bloodstream. So we'll collect the tumors in the blood and then use the streptavidin pull down to isolate the EHE specific proteins and then identify what they are by mass spec. In parallel, we have a mouse that will basically attach this biotin to every single protein that it secretes from all organs of the body. And so this will allow us to identify the proteins that are normally secreted by the mouse into the blood. And we can sort of eliminate those from our list so that we're focusing only on the proteins that are made in high abundance by EHE cells, but not other cells in the body. And that makes this biomarker EHE specific. And then through our collaboration with Sandra Pasquale's lab, which is also funded by this grant, we will then use access to EHE patient blood samples and healthy donors to identify or to validate the markers that we identify in this approach as being EHE specific in actual human patients, allowing us to do some of the things that I mentioned on the previous slide. And ultimately, what we would like to do is then use this same model to test whether treatment with drugs, so if we can make these EHE tumors in the mice, treat the mice with drugs that we know will have an effect on the EHE growth, and see whether the levels of those biomarkers that we've identified go down. And that will tell us that these are good readouts for this drug working. And then we can also do the same thing to look at the progression of the disease by taking levels of the biomarkers at different points in the growth rate of the tumors and asking whether transition to an aggressive form of the disease is it correlates with an increase in a biomarker, and that might help us predict in patients whether or not they're likely to progress to a more aggressive form of disease. And so those are the immediate goals of the project. We've just started this. The grant has just been awarded, and so Sarah's been working very hard, and she's got the first experiment done showing that we can in fact label our tumor cells with this, or put the enzyme in the tumor cells to label the proteins. This is just a Western blot showing the biotinylated proteins from the tumor cell lysate, from the tumors themselves of these mice, with all the biotinylated proteins. And more importantly, she can enrich the biotinylated proteins that the cells are secreting into the bloodstream. And so these are isolated specifically from the blood, and we can see a lot of biotinylated proteins there. And so we're ready now to hopefully send, Sarah's just doing some more control experiments to make sure this is working as we expect. And we'll be ready to send out the first set of samples to look and see if we can identify EHE specific biomarkers using this approach. And I will stop there and thank all the members of the lab who've been working on this in purple here, all of our collaborators here, both at my institution and across the world, and obviously the EHE Foundation and other sources of funding throughout the years for these EHE projects. And turn it over to whoever is next.
Thank you much. It's really exciting to see all the worldwide collaboration on these projects bearing fruit.
Hello, everyone. I'm Gillian DeWane, and I am in Dr. Munir Tanas's lab at the University of Iowa. And I'm going to be talking about my project looking into combination therapy approaches targeting genomic and signal transduction vulnerabilities in EHE. So the Tanas lab primarily studies EHE, which is a vascular sarcoma that's characterized by either the TAS-CAMTA1 or the YAP-TFE3 fusion proteins that are the main oncogenic drivers of EHE. The fusion proteins act as transcriptional co-activators by binding to the TEAD family of transcription factors to drive the oncogenesis of EHE. So EHE is commonly found in the liver, lungs, and bones, and treatment for EHE patients can include surgery, radiation, chemotherapy, or targeted therapy. So a number of different medical therapeutic approaches have been tried in EHE over the years, including rapamycin derivatives, topoisomerase II inhibitors, and now some TEAD inhibitors, but treatment options are still limited. So my project in the Tanas lab has focused on investigating potential targeted therapies in EHE to ultimately try to improve treatment options for EHE patients. So to approach this question, we looked at some transcriptomic data from RNA-seq studies performed on our engineered cell lines that express the TAS-CAMTA1 or the YAP-TFE3 fusion proteins. And pathway enrichment analysis was performed on these cell lines, and we looked at the top cancer-related signatures that are activated by the fusion proteins. And one of the pathways that was activated by both fusion proteins appeared to be the PI3K-AKT signaling pathway. So then to validate this transcriptomic data, I looked at activation of some of the proteins in the PI3K-AKT pathway. So looking at phospho-AKT and phospho-S6 and compared them to empty vector controls. And we saw a modest increase in AKT and phospho-S6 activation. So together, these data suggested to us that the PI3K-AKT pathway may be a potential pathway to target for treatment of EHE. So in addition to our engineered cell line systems, we've collaborated with the EHE Foundation to generate these primary extended EHE cell cultures from patient tumor samples that we received from the EHE Foundation. So after these cell cultures were made into cell lines, we were able to validate that these cell cultures contained the fusion protein. And then we also validated doing functional studies to make sure that they were representative of EHE. And we performed similar transcriptomic analysis on these EHE cell cultures as we did the EHE engineered cell lines that we utilized before. So again, we performed bulk RNA-seq on these two of the EHE cell cultures that we generated, shown here, and some of the significant cancer-related pathways that appear to be enriched in the EHE cells are shown here. So similar to our engineered cell lines, we saw activation of the PI3K-AKT signaling pathway, suggesting again that PI3K pathway is important for the fusion protein-driven progression of EHE. So again, I validated these transcriptomic data analyses by looking at activation of some of the proteins in the PI3K-AKT pathway in two of the EHE cell cultures here, and saw an increase in phosphorylation of AKT as well as phospho-S6 compared to a human pulmonary artery endothelial cell line as a control. So together, these data validated our engineered cell line model of EHE and corroborated our findings that the PI3K-AKT pathway is important in the pathogenesis of EHE. So with this data, we hypothesized that targeting mTORC1 with everolimus in combination with targeting TEAD with IK930 will decrease tumorigenesis to a greater extent than either inhibitor alone. So to test this hypothesis, we treated our engineered cell lines expressing the fusion proteins with various concentrations of everolimus, which again targets mTORC1 in the PI3K pathway, as well as IK930, which targets TEAD either alone or in combination with one another. And we plated them in soft agar to look at anchorage-independent growth, which is the main oncogenic phenotype of EHE. We then assessed whether these two compounds worked synergistically with one another and showed that at 10 nanomolar everolimus and 100 nanomolar IK930, these compounds showed synergy and significantly decreased anchorage-independent growth better than either compound alone at these concentrations, which is shown in these graphs and photos. Additionally, we tested these compounds in combination in some of our EHE cell cultures and showed that with combination therapy, everolimus potentiates IK930 in soft agar in the EHE cell cultures. However, even though we saw a decrease in colony formation with the combination treatment, it still did not completely abrogate anchorage-independent growth, indicating other mechanisms may need to be targeted in EHE. So in parallel to these dual combination studies, our lab performed a high throughput drug screen to identify other potential targets in EHE. So briefly, we plated NIH3T3 cells expressing the YAP-TFE3 fusion protein in low attachment plates and allowed them to form spheroids. Once spheroids were formed, they were treated with 10 micromolar of a compound in which 1,000 FDA-approved compounds were tested. The spheroids were allowed to grow for several days, and then the spheroids were imaged and analyzed. So the screen was repeated twice, and compounds that overlapped in both rounds were tested for a dose-dependent response, looking for a 50% decrease in spheroid size compared to the control. So from this screen, 6 compounds were significant in decreasing spheroid formation in 3T3 cells expressing YAP-TFE3, 4 of which were topoisomerase II inhibitors. So a little background on topoisomerase II. Topoisomerase II is an enzyme that has an alpha and beta isoform that have similar but not identical cellular functions. Topoisomerase II is involved in regulating chromatin architecture and resolves supercoiling in the DNA during replication and plays several roles in regulating the transcription of genes. So the data from the screen resulting in 4 out of the 6 significant compounds being topoisomerase II inhibitors suggested that topoisomerase II may have an important role in the pathogenesis of EHE. So to validate the results of the screen, we plated both NIH3T3 cells expressing YAP-TFE3 as well as TAS-CAMTA1 and soft agar to measure anchorage-independent growth and treated them with various doses of teniposide, which is one of the topoisomerase II inhibitors that was identified in the screen. And then we showed in the 3T3 cells expressing the fusion proteins that at higher concentrations of teniposide, teniposide completely inhibits anchorage-independent growth. And then since 3T3 cells are a mouse cell line, we wanted to validate these findings in our human engineered cell lines. And we saw similar results showing a decrease in colony formation with increase in concentrations of teniposide, but only a complete abrogation of colony formation at high doses. And in addition to this drug study data, we performed various genetic knockdown studies in these cell lines that targeted both topoisomerase II isoforms and saw a similar decrease in colony formation with knockdown. So together, these data further supported our working model that topoisomerase II is a critical player in YAP-TFE3 and TAS-CAMTA1 driven anchorage-independent growth, which is the main oncogenic phenotype of EHE. So altogether, with this data, we hypothesized that treating the cells with a low dose of teniposide in combination with everolimus and IK930 would further decrease the anchorage-independent growth phenotype than any inhibitor alone or in combination. So to test this, we performed similar experiments as before and treated our engineered cell lines with various concentrations of teniposide here, as well as our synergistic concentrations of everolimus and IK930. And these results showed a dose-dependent decrease in colony formation with complete inhibition of colonies at higher concentrations of teniposide. Similarly, we did the same experiment in our EHE cell cultures and saw inhibition of colony formation at lower doses of teniposide in both of these EHE cell cultures. So these results suggested to us that a physiologically relevant dose of teniposide in the nanomolar range could be used to sensitize EHE cells treated with everolimus and IK930 to eliminate this anchorage-independent growth phenotype. And then after examining this triple drug combination in vitro, we wanted to test its efficacy in vivo. So to do this, we collaborated with Dr. Sandro Pasquale in Italy, who gifted us his EHE PDX cell line model, which is an aggressive form of EHE, and we utilized them for a mouse drug experiment. So briefly, we injected the EHE PDX cells into NSG mice, allowed the tumors to become palpable, and divided them into 6 groups. We treated the mice bearing these EHE PDX tumors with vehicle, with everolimus alone weekly, which corresponds to the red arrows in the graph here, IK930 daily, everolimus and IK930 in combination, teniposide alone weekly, which corresponds with the orange arrows here in the graph, as well as the triple combination of all 3 compounds. Mice were treated for about 7 weeks, and after the initial growth period, the triple combination group, which is designated by this blue line here, the tumor stabilized for the remainder of the experiment, while all other groups kept growing. Importantly, the dual combination group, which is this purple line here, and the triple combination groups grew slower than the vehicle group up here in black, as well as all the monotherapy groups. And the triple combination group had significantly smaller tumors compared to the dual combination group by the end of the study. So these in vivo results suggest that potentially cycling in teniposide for treatment with everolimus and IK930 may keep EHE tumors from growing. So in conclusion, the results from my project showed that through 2 unbiased approaches utilizing bioinformatics and a high throughput screen, we identified several pathways and compounds to use as potential targets and treatment for EHE patients. I also showed that mTORC1 in the PI3K pathway and TEAD inhibition synergistically decrease anchorage-independent growth in cells that represent EHE, but does not completely abrogate this phenotype. I also showed that topoisomerase 2 inhibition appears to sensitize EHE cells that are treated with mTORC1 and TEAD inhibitors, both in vitro and in vivo, to a greater extent than just mTORC1 and TEAD inhibition alone. And then future avenues in identifying other therapeutic targets could be to examine other significant pathways that are important in EHE tumor progression, as well as other significant compounds to treat patients with EHE. And then I wanted to acknowledge everyone who assisted with this project in the TNAS lab, especially Nick Scalora, who generated some of the EHE cell cultures with me, as well as acknowledging the EHE Foundation and the funding that I received to help push this project forward, as well as the Pasquale Lab and Dr. Sandra Pasquale for providing the EHE PDX cell line for these in vivo mouse studies. Thank you.
Thank you so much. Very exciting work in a very short amount of time. Excellent. We have one more presentation in this second session. I'm going to invite Dr. Pobbati, who's going to be speaking, and from the Cleveland Clinic, who's going to be speaking about targeting the oncogenic transcriptional complex in EHE.
Everybody able to see my slides and hear me?
Your audio is coming through great.
All right, cool.
Slides just need to shift to the...
All right.
Perfect.
All right, thank you very much for inviting me to this great conference, and I always enjoy presenting here. I learn a lot as well, and it also provides me an opportunity to address patients directly. So I'm Ajaybabu. I'm an assistant professor at Cleveland Clinic Research. I work in Rubin Lab. So I'm going to tell the same thing that other people have said, that EHE is caused by a fusion gene. I'd like to draw this analogy to like a minotaur. What this fusion gene contains is that it combines two properties. One property is from TAS, and the other property, it gets it from the CAMTA1. The analogy of a minotaur is that, like, minotaur has can think like a human, but it has a body like a bull that makes it a lot more powerful. So the ability of it to turn on genes, it gets it from the TAS portion, and the ability of it to stay in the nucleus, it gets it from the CAMTA1 portion. So previously we talked about, Kieran talked about the Hippo pathway acting as a brake. So the CAMTA1 overrides this brake and keeps the TC in the nucleus. And even though this is a monster, this is a minotaur, it cannot act alone. It needs company of a transcription factor called as TEAD, which we also discussed by several speakers in the previous talks. So what this oncogenic complex does is that they pair together and turns on genes. These are the genes that promote cancer. So the ability to stop TEAD from acting has gained a lot of traction because there are small molecules that bind to TEAD. So TEAD has some pockets that allow small molecule to bind. If you want a small molecule is something like drugs that you take through orally. So these are the drugs that needs to bind to our target. And in this case, the target protein is TEAD. And what it does is that it binds to TEAD and prevents TC from interacting with the TEAD. Actually, in the literature, most of the people are talking about the parent protein, which is the YAP and TAS form. And that is what research is mostly done about. These small molecule binds to TEAD and prevents YAP and TAS from interacting with TEAD. And what happens is that this acts as a disruptor of this interaction. And once you disrupt this interaction, this complex loses its ability to turn on genes. So there are two ways that that you can target TEAD. One is using a central pocket, and the idea of a central pocket targeting also came largely also from our work, previous work, and this is targeting approach has gained traction, and you now have several central pocket targeting drugs. A good example is Vivace Therapeutics drug, but there is also a pocket on the surface, and this is exploited by Novartis. So there appears to be like two different ways of attacking TEAD. And here I'm going to show an example. We've used a lot of surface binding TEAD inhibitors, and this is the IAG 933, which is a Novartis drug. Like I said before, the Novartis drug has the ability to disrupt the YAP-TEAD complex and also the Cas-TEAD complex. But the question is, does it also retain the ability to disrupt the TC-TEAD complex? And this is shown here in the form of a pull-down assay. Basically, I try to keep it high level as possible so people can understand. So what we are doing here is we are pulling down our monster, which is the TC, using an antibody and looking at TEAD when it comes with it. So that's what you see here. When you pull down your TC, TEADs come with it. But when you add the disruptor like IAG 933, the TEAD is no longer coming with it. So the mechanism of action is also applicable for the TC-TEAD complex. So now that we have disrupted the TC-TEAD complex, can we check whether it loses its ability to turn on genes? So some of the genes that people use in this study are CYR61 and AMOD-L2. So this experiment here allows us to understand whether when we increase the concentration of this drug, in this case IAG 933, whether this oncogenic transcription complex loses its ability to turn on genes. And that's what we are seeing. So the red down arrows downward, meaning that this complex is losing its ability to turn on genes. And what does that mean for cancer cell growth? What we are seeing is that as you increase the concentration of these TEAD inhibitors, the cells are no longer growing. But there is an important caveat here. These cells are like frozen in time, but they are actually not dying. One of the things that we have noticed in vitro is that the surface binders are faring better than the central pocket binders. Obviously, I've used a very old candidate, MGHCP1. There are advanced molecules here, but the trend seems to be the same. So at this juncture, I find it important to compare in a clinical setting, what is the status? The surface binder, the only one that is brought to the clinic is IAG 933. Ours being a rare cancer, there's only 9 patients registered. But one of the things that they try in a clinical trial is to assess whether the compound is safe, whether the compound is tolerable, and so on. Unfortunately, the IAG 933 failed on the primary endpoint, which is the tolerability, because it has something called as a QTC prolongation, or in other words, patients who are taking IAG 933 seems to have heart problems. But nevertheless, the EHE patients respond really well to IAG 933. If you look at the disease control rate of about more than 50%, that means more than half of the patients respond to IAG 933. But one important thing that we understood from this thing is that the tolerability effect is very likely to be an off-target. That means inhibiting TEAD is not causing this effect. Why do we say that? Because we have another study which has been released in the CTAS, and Dr. Chen touched upon it, is the VT3989 data. So both compounds inhibit TEAD, but the off-target effect, the tolerability effect that you see happens only in the case of IAG 933, not in the case of VT3989, suggests that what we are seeing is probably an off-target effect, which is a great news because this effect is not caused by inhibiting TEAD. And even for VT3989, the patients taking this drug are responding, which is a great news. So IAG 933 project has been shut down, but it has inspired several companies. So what I'm showing here is that many companies have taken these scaffold, the Novartis scaffold, and have designed surface disruptors. But given that these surface disruptor, especially when you use a Novartis scaffold, you seem to have other off-target effect, I suspect that these other compounds may also carry the same effect because they are mimicking the Novartis scaffold. But interestingly, these two companies, the Amphista and Biactica, are something that we need to watch out because they're using a different approach. They are not using disruptors, but what they are using it as degraders. So these compounds have the ability to deplete TEAD in the cells, and we don't know what the properties of these compounds in the clinic is going to be. So it'll be interesting to see if these companies brought their small molecules to the clinic. So we also have a big effort in generating a novel T surface disruptor. So I'm a co-project leader here. The main PI is Sean, and Brian is the co-applicant PI. So we have a really novel, distinct scaffold from Novartis. It's been very difficult to generate a scaffold like that. That is a surface binder. In fact, we started with binding compound at the triple digit micromolar range. We are now in the low nanomolar range. We have improved the potency by over 3,000 fold, but nevertheless, it took us about 700 small molecules that need to be synthesized to get that. This is the whole team that we have. I'm really proud of being part of this team because we have found a scaffold that mimics nothing like Novartis, and this combines multiple multidisciplinary expertise here. So this is the current status. So eight companies tried to bring T inhibitors to the clinical trials. 3 of them backed out. But the good news is that 2 companies, VT3989 is entering phase 3, and ODM, as already previously mentioned, is entering phase 2, which is a great news for the community. So I'm going to shift gears here. So for the first part of my talk, I talked about targeting the TEAD. What about targeting TC? Because they are also part of the transcription complex. But the trick or the difficulty in targeting TC is that it does not have any verified pockets like like TEAD. So if you don't have pockets, how are you going to target them? Being this many years in drug discovery, one thing I've noticed is that there is nothing like that cannot be targeted. You have to think harder, think out of the box. So we came up with this idea of a phenotypic screen where we are looking at TC, not at the single molecule level as Kieran showed, but this is at a whole global level. What you're seeing are dots, nucleus, and this is where the TC is predominantly located. And what we did is called as an approach called as drug repurposing approach, meaning that here in this project, we are not synthesizing a new drug like the TEAD project, but we are trying to repurpose the drugs that are already present. In our screen, we had about 2,000 FDA approved drugs, or drugs that can be turned in, or small molecules that can be turned into drugs. So that we can push this project faster. Cutting a long story short, this has been published in Clinical Cancer Research a couple of years ago. So starting with 4,000 compounds, we narrowed down to 5 compounds that clearly reduce the amount of TC in the nucleus. That's the purpose of the screen. Look for compounds that reduce the amount, that reduces the monster in the cells, so that this is the monster that's creating so much havoc. We want to reduce its level in the nucleus. So these are the 5 drugs that we identified from the screen in an unbiased manner based on IC50 values. We focused on dinacyclib. So dinacyclib is a cyclin-dependent kinase inhibitor. We don't have to go into detail of what this function is, but it can inhibit 4 cyclin-dependent kinase, 1, 2, 5, and 9. So similar approach. In this case, what we are doing is we're completely depleting TC from the cells. There is another issue because this compound is not just decreasing TC, but also decreases other stuff. So we got to bear in mind what we are seeing. But one thing that we are predominantly seeing is that even here, the ability of this oncogenic transcriptional complex to turn on genes is dramatically decreased. And again, we are looking at what happens to the cells when your ability to turn on gene is reduced. In the cells, what we are seeing is that this is even more potent. What is seen in the orange is the cyclin-dependent kinase inhibitor. It's much more potent than the surface binding inhibitor, and it's also even more potent than the central pocket inhibitor in comparison. But interestingly, when you add these cyclin-dependent kinase inhibitors, the cells actually die. This is in contrary to what we use as a TEAD inhibitors. There, the cells are actually not dying, but they are growth inhibited. When you have a cyclin-dependent kinase inhibitor, the cells are actually dying. And here is an example where we tried to narrow down. Like I said previously, DINA inhibits 4 of these cyclins, and we wanted to know which of these is important for its function. And like I said, when you add these compounds, the amount of the monster in the cell, which is TC, dramatically reduces. And you can see that here in a form of a blot. So TC disappears when you add dinacyclib. The only compound that makes TC disappear is the AZD4573, which is a selective CDK9 inhibitor. So this is a project, this inspired us to investigate it at a larger level. And this is funded by EHE Foundation. So thank you for funding it. And everything that is shown in red are very selective CDK9 inhibitors. So we are now prioritizing CDK9 inhibitors using several assays, including mouse models, and want to come up with one molecule that could potentially act as a molecule that can be used to treat EHE. So I want to summarize here. So the oncogenic transcriptional complex is the TEAD and the TC. Both of them can be targeted. You can use the TEAD inhibitor. You can use a CDK9 inhibitor for targeting TC. I know people can get demotivated when they see pharma shutting down TEAD inhibitor programs. Don't get demotivated. This is the norm because it is called as a valley of death. 90% of the molecules coming from entering phase 1 will not go to the clinic. Unfortunately, that is the norm. But please be hopeful because you have two small molecules that are progressing, the VT compound and also the Orion compound. So there is a lot of hope for it. And you should also think about other ways of targeting EHE. We talked about, Dr. Chen nicely mentioned about sirolimus, which is also work from Stacchiotis group, and Silvio, and also we talked about, John Lamar also talked about statins. So I'm taking what David used to say. It's always good to have more shots on the goal. He didn't patent protect this phase, so I'm using it. And none of these would be possible without a team that I had. So I work in Rubin Lab, and I also collaborate intensively with Sean Stauffer. So this is my team, helped me a lot in this project. And this is the phenotypic screen. And none of these would have been possible without generous funding from these sources. Thank you for your attention.
Thank you so much for the update. If I can welcome back the session 2 participants, and I believe we also have Dr. Tanash joining the discussion here.
Oh, I have to stop sharing. Let me see. Oh, yeah.
Excellent. And Christy, you want to kick us off with some questions?
Great. Yes, thank you. Thank you so much for these wonderful talks. They have sparked so much really interesting discussion here in the chat and in the question session. I'm going to start off with a question that's really for Dr. Lamar, but I think everybody may have some input on this regarding statins and how the drugs interact with other medications that might be used to treat EHE, specifically sirolimus and everolimus. You mentioned that statins should not be used in combination with sirolimus, but both sirolimus and statins are kind of in groups of drugs that behave similarly. Can you talk a little bit about whether that's true for sirolimus and everolimus, and if that's true of all statins?
So I'll leave the sirolimus everolimus question for Munir, but I believe they have similar mechanisms of action. For the statins, I think we're largely targeting a different pathway here. At least that's what we think is happening in the EHE cells. I think similar to what Ajay showed, we think what's happening is that the statins are causing TASCAMTA1 to leave the nucleus, at which point it's getting degraded. We see similar effects to what he showed. I didn't show that data in the interest of time, but we see the protein go away. It's probably also impacting other important proteins in the cell. So many of you have probably heard of the trametinib trial that was done in EHE. The statins are also reducing, are sort of slightly targeting the same thing that the trametinib is by inhibiting the an enzyme, by inhibiting the activity of those proteins, the same ones that the trametinib is. And we know that in these same cells, trametinib has an effect on the viability as well. And so I think there are probably different pathways that in the statins probably hitting a couple different things, maybe not as potently as the direct targets will, but it's providing multiple benefits, we think, in the context of EHE. Your first point about combining it with other therapies, again, yeah, you can't combine it with sirolimus. That's definitely not something you should do. And I think just in general for patients out there, especially for something like statins where you may be on it already for cholesterol regulation, you want to let your doctor know that you're on that, and they will be the ones to look up, potential dangerous interactions and not only dangerous interactions, sometimes drugs can sort of inactivate or prevent the function of other drugs. And so you don't want to adversely affect a treatment that might be working by putting something else on that you think might work. And so your doctor should know kind of how to handle that and which drugs to avoid for interaction reasons.
Thank you. Dr. Tanas, did you want to add a little bit to that?
Yeah, I mean, I think in terms of differences between everolimus and sirolimus, they're very similar in terms of mechanism of action. Everolimus has a shorter half-life, but is more bioavailable. So you can make the argument either way. I think in our studies, in our mouse studies, we have a dosing regimen that appears to, that we become comfortable with and the mice tolerate. And so that's the primary rationale. But you can probably almost certainly substitute sirolimus and, presumably see similar results. But you would have to do the experiment to confirm that.
Got a question for Dr. Pobbati. So when we're talking about these CDK9 inhibitors are, essentially directly attacking the minotaur more than so much focusing on TEAD. Can you speak a little bit about, the pros and cons of that approach? One of the questions is always when we're inhibiting TEAD is sort of used normally in cells for normal functions, so you worry about side effects. Would that potentially have less side effects? What do you think about combinations with TEAD inhibitors? What are your thoughts about pros and cons of TEAD inhibitors versus going upstream to the minotaur?
Right. So that's a really great question. And at the back of my mind, I always worry about it. I wish we hadn't identified like just CDK9 inhibitor, because most of these screen are somehow targeting CDK9. The problem with the CDK9 inhibitor is that it is inhibiting actually transcriptional process. So your TC, the monster, is not the only thing that is going down. There are other proteins that are also going down. So it turns off multiple things. And that can become a problem, because if it turns off something that is really important for your body to function, your therapeutic index is going to narrow. And that is something at the back of my mind we should always be concerned about. But none of this, we would know whether it will work or not until or unless we try it in patients. So that's the sad reality. Like, is the therapeutic window good enough so that it could be used as a drug? But nevertheless, we got to try. But with regards to the combination therapy, that seems to be like a good way to go, because in multiple research studies, I've shown that also from John's current presentation, shown that if you combine both these drugs together, you are left shifting the curve. The cells are dying a lot more at a much lower dose than the dose required for the individual compound. So you can call that synergy, you can call that additive effect. And what this could potentially do is that, you could try to minimize those side effects. Again, this is all theoretical. You got to test it. That's the real test. But the hope is there. So when you combine these two compounds, you are left shifting the curves, you're using the drug at a much lower concentration, and overall, you're trying to improve the therapeutic index.
I have a related question, and I think all of you can speak to this to some degree, but maybe Dr. Tanas, you can start us out. So when you're looking at these drugs in a research environment, most of the time you're testing them on a cell line, at least to start. And then there's this next question of, as we're combining drugs together, how do they interact with each other? And then what are the effects on the person taking the drugs? What are the limitations of the tools that you have in being able to ask that question in your lab environment? Are you able to ask that question in your lab environment and learn how the drugs interact with each other and how they will potentially impact side effects in patients?
Yeah. Yeah, and as you alluded to, in the initial stage in screening for compounds or identifying compounds that will work together is usually some sort of a screen or cell culture based system that may recapitulate portions of the actual human disease, but not entirely. And so we validate it in mouse models and tissue samples. And so in terms of efficacy, I think the more model systems that you can use to corroborate your findings, the better. And then along the same lines, in terms of looking at deleterious effects of the combination. I think the same principle applies. And so, you can look in cell culture systems and see, you can use normal cells, kind of primary cell cultures, and compare them to your cancer cell lines and see what the therapeutic window might be for a certain compound or combination of compounds. And you can do various toxicology type studies in your mouse studies as well. And so the most straightforward thing people use is just weigh the mice, and throughout the course of the study, we've done that, and you can determine whether at a macroscopic kind of gross level, is there a deleterious effect of that combination on your mouse model? And you can do, on a pathological level, you can examine various organs. A lot of times there's known toxicities of this compound. So you can do, for example, a full necropsy and at the end of the study, and we've done that to determine whether there's an off-target effects on various organs. And then you can do, formal toxicology type experiments on you can look at serum levels. There's a lot of different ways that you can, in your mouse studies, that you can kind of address that question. So you can approximate it. I think you can get a pretty good idea before you go into a phase 1 clinical trial, whether that, what some of the side effects might be. But in the end, I think once you've solidified your preclinical data as well as you can in your models, what it actually does in patients in phase 1 clinical trials are the most telling. So you have to see how the patients tolerate it at that stage, which is the whole purpose of a phase 1 clinical trial.
Sure.
Any other thoughts from the group or final comments from this session before we move on to the next session?
Yeah, I guess I'll just make a comment based on what Munir just said, that, there's this push, especially in the United States, to kind of move away from animal models. And I think what he said and what you see today is that those still have a very important role, especially in drug discovery. And so, while it might be possible to start coming up with other models, we really still need those animal models. Until that changes, it's just something that I think the general public needs to be aware of in these sort of political times, I guess I would say. So there is an important role for that. We can't understand drug interactions and toxicity without having a living organism to test them in. And mice are a good way to do that now. And so while we can try to minimize how many mice we're using, we still need them if we're going to find new treatments for patients.
I entirely agree. And in addition to the mechanistic insights, there's a safety argument to be made too. I think you definitely, I would say, improve the odds that you're not going to have a deleterious effect in a phase 1 clinical trial if it's extensively studied in vivo. And so I entirely agree with John's comments, and we need to continue these studies for sure.
Yeah, I echo that too. I mean, we don't want humans to be the guinea pigs. The animals should come in between. We don't want to hurt them, but we want to know how toxic the drug is, whether it's tolerated or so. And they still are the best way to learn.
Thank you so much for that.